I'm going to talk about, I've changed my title very slightly, using sequence covariance to make inferences about protein structure and function. So this talk is going to be quite different, I think, from the talks that have gone before, in that there will be a bit of biology. I'm going to try and explain everything, but if you're at all confused at any point, please just ask a question, put your hand up. I'm more than happy to be interrupted. Okay, so sort of made an outline of the talk. The first part is going to be sort of the result, in the sense that we're going to show that you can use sequence covariance, and I'll explain what that means. But you can use sequence covariance to give accurate prediction of 3D protein structure. And that's sort of subject to several caveats, you know, sort of. Oh, sorry. Okay, all right. So the first part of the talk, I'm going to sort of show you that sequence covariance can give accurate prediction of protein structure. It's 3D protein structure. And that's sort of subject to several caveats. So we're going to use lots of sequences to do this, and we need those sequences to be well sampled. So I'll say a little bit about that. And then I'm going to sort of move on to at least asking some mathematical questions. So, you know, how well does this work if there are fewer sequences? Certainly the prediction quality gets worse. And more to the point, how do we know if there are enough sequences? How do we know if we have enough data to do this? If you give me a data set and I make a prediction, I sort of have no idea if it's right or not, if I don't already know the answer. And I'd like to sort of have some idea of how well I can do just by looking at the original data. And you know, if there aren't enough sequences to actually predict the whole protein 3D structure, then can I still obtain some reliable information from that data? And to sort of try and get at those questions, or to start getting at them, I'm going to use some ideas from random matrix theory. In particular, I'm going to ask, can we use spectral analysis to distinguish useful signal from the sort of noise that we get because we're so undersampled? So first, I'll start off by sort of introducing the problem and this idea of sequence covariance. Oops. So, hey, this slide shows a linear amino acid sequence. This is what the unfolded polypeptide chain looks like. So in any sort of living organism, when a protein is made, it's made as this unfolded linear sequence. It might start to fold up as it's being made, but it comes out as a chain. And there are amino acids, you can't see them here, but these sort of separate boxes indicate individual amino acids. And those are numbered according to their sequence position. We just number through the sequence. And essentially the idea is that this polymer self-assembles into some kind of unique 3D structure, and it does that the same way every single time. And then those monomers, those unique 3D structures, interact either with copies of themselves or with other proteins to make functional complexes. Right? But somehow the problem we're interested in is getting from this linear chain of letters of amino acids to a folded functional protein molecule. I'd like to be able to predict from just this sequence, right, what the structure and function of the protein are. That's sort of the protein folding problem. Oops, sorry. So each protein is constructed as this chain or string of amino acids. There's 20 different amino acids, so we've got sort of 20 different states the system can be in. And the order of these amino acids is really important, right? So really, we'd like to find a probability model for the sequence of amino acids that corresponds to any given protein of interest, right? So I'm interested in hemoglobin, I'd like to be able to make a probability model for this sequence. So I'm envisaging that I'm going to have a model that has sequence positions as the variables. So these are these different positions in the sequence, 1, 2, 3, up to L. And each of those positions can be in one of these 20 or 21 different states. Okay, and I'd like to actually make a model. You know, I'm going to have as a function of these variables that will tell me which sequences belong to that family. Okay, so that's sort of the problem that I'm interested in. And essentially what's interesting is that we now have many, many versions of these sequences. Right? We've sequenced lots of different species, we've sequenced lots of samples, you've all heard about the falling cost of sequencing, so we've got all of this data. So essentially we've got many versions of the sequence for any particular protein we're interested in. So say this is hemoglobin, we have lots and lots of different versions. Each line in this plot is a version. of that sequence. And so we'd like to use that data to actually parameterize the model for this protein sequence. And you can see in this particular sample I've shown you, there's not actually that much variation, right? Quite a lot of these columns are really, we call them quite conserved. They stay the same amino acid and, all the way through this set of species. But this is a very small set of species. We have thousands and thousands of these sequences, and we'd like to be able to use that data in order to infer a model. So essentially, these different versions of the sequence record the outcome of millions of evolutionary experiments, right? So, as species evolve and change, mutations occur, and they're either accepted if they allow the protein to still work, or otherwise that particular instance doesn't survive. So proteins actually perform lots of different functions. You've got ligand binding, 3D structure of the protein. They bind to other proteins. They carry out specific tasks. And those functions are generally sort of controlled by groups of amino acids, right? So we'd like to understand in the sequence which groups of sequence positions actually control these functions. We're going to be able to engineer these proteins and sort of, change them so that they do what we want them to do. Then we need to understand what the relationship between that linear sequence I showed you before and these different functions actually is, right? And we'd like to basically invert the sequence data to identify these constraints, right? So essentially, at the very end of the day, we'd like to get to design, but for now, we're interested in just trying to understand, what kind of model might be appropriate to actually sort of model this sequence data. So for a long time, people have looked at first-order models, and by that I mean models that just involve linear terms, right? So essentially, you've just got terms that have, a single sequence position in them, and we're making the assumption that different sequence positions evolve independently of one another. So we have no interactions. There are no higher-order terms. People have made hidden Markov models with this assumption, and they've actually been very successful. They've been very useful in that they've allowed us to build large protein sequence alignment. So we have these protein families that we make with hidden Markov models, and they contain thousands and thousands of sequences, and essentially, those are very useful for lots of different purposes. But sort of the key question is, do they tell us anything? Do these models tell us anything about protein structure and function? And in that sense, they're a little bit limited, right? They only involve individual sequence positions. We sort of know that sequence positions that don't change much across evolution are generally quite important for function, but that's about all you can say. And so, this is the structure of that unfolded polypeptide chain. Even that sort of first-order cystic conservation doesn't actually really tell us anything about the 3D structure. So basically, in order to get any further with this question, we're going to have to include some kind of interactions between these sequence positions. We're going to have to put higher-order terms into our model. And so to start with, we might as well consider second-order terms. And there's a sort of toy model up here which indicates why these sort of second-order terms involving two sequence positions might be important. And this just sort of illustrates charge pairing. So here, this is a protein, this blue object's a protein. These are two particular amino acids that happen to be oppositely charged. And if you have a mutation that changes one of the charges, then you can imagine that might stop the protein from folding up correctly. But if you have a second mutation that sort of flips the other charge and restores that charge pairing, then you can imagine having a functional double mutant. So the idea is that in the sequence database, you either see this combination, right, or this combination. But we'd never see this interim combination because this is not a functional folded molecule. Okay? So essentially, we're sort of asking, how do mutations at different sites depend on each other? And what constraints on the mutations that can be accepted are induced by the requirements of structure and function? And so this just sort of reiterates that point I made, that we'll either see D and K, this wild-type version, or R and D, this double mutant. And so if we look at the columns of this little toy sequence alignment, you'll notice that there's a strong correlation between the first column and the last column, and that's the correlations that I'm talking about. We're literally going to take these sequence alignments, and we're going to look at the sequences of letters that are in different columns, right? And we're going to measure their correlation. So this is the actual alignment that I showed you a little while ago, and you might sort of be thinking, well, that's a little bit implausible, but I can actually find columns even in this small data set where there's an almost perfect correlation between where they change in this particular set of sequences, right? And it could be that these two sequence positions are actually interacting with each other, or it could be that I've just picked a very small set and I got lucky, right? But that's the kind of signal that we're looking for. Okay. What we'd really like to do, of course, is be able to sort of identify that signal by looking at correlations in these large sequence alignments and be able to use that to make predictions or inferences about pairs of sequence positions that are close to each other in the folded 3D structure. Okay, so we want to invert the correlations that we observe to provide information about this 3D structure. So essentially the problem is, I mean, these sequences are very easy and cheap to obtain, but actually finding the 3D structure of a protein, I mean, it's possible experimentally, it's somewhat routine, but it's still quite expensive and difficult to do. Right? So if we could predict the structure from sequence, then we'd be in much better shape. So what are we going to do? We're literally going to start with really large sequence alignments. So this alignment has 4 sequences in it. I'm going to be using alignments that have, say, 10,000 or 20,000 sequences in them. They'll typically have something like 300 or 150, 300 sequence positions. So that's the variables. And we're literally going to build a correlation matrix. Okay, so we're going to take every single pair of columns, we'll compute something like the mutual information and build a correlation matrix. And then we're sort of asking, does that tell us anything about the sort of structure or function of the protein? Okay? And then specifically, are the most correlated residue pairs close in tertiary or 3D protein structure? So let's test that. Right? This is just using mutual information. This is a contact map of a protein. So this is actually a protein structure. This is the answer. Right? This is an experimentally determined protein structure. And this is just a binary matrix that represents that structure. It's a bit like an adjacency matrix. There's a 1 wherever these two sequence positions are close to each other in the 3D structure. Right? So you can see the diagonal is all 1s. That's because every position is close to its neighbors. And then, you know, in this protein, sequence position 160 is close to position 110, or whichever one that is. Right? So this is just a matrix that tells us the structure of the protein. We'd like to be able to predict this. And so on top of this, I'm going to put the residue pairs that have the highest mutual information values. Okay? And if this is all going to work, then the red point will fall directly on top of the blue points. Okay? So this is just the top 150 values, and you can see they're sort of concentrated on the diagonal. There are some off the diagonal. If I put some more points on this plot, we start to see that really this isn't working in any sense at all. Right? We've got these large sort of blobs of red or sets of points that all have high mutual information values, and they're not really overlapping with most of these blue points that are contacts in the 3D structure. So this is kind of disappointing. But if we look more closely at this, this result, it gives us a clue about what's going on. We sort of zoom in on the top corner of that result, and we think about a toy model. We can sort of see we have these sort of blocks of red, right? Or these solid lines of red, so these chains of amino acids that have high mutual information all with a single position. So if we think about this single model, well, in this model, you know, this K is close to the D, right? So we'd expect the K and the D to have high correlation if our whole premise is correct, right? But, you know, obviously the K is also close to this E and the D is close to this R. So if our premise is correct, right, then we're going to expect a result that looks something like this. The only problem is that we're talking about correlation here, right? So these are columns of amino acids, and this correlation means that every time the E changes, this column changes, the K column in the alignment changes. And every time the K column changes, the D column changes. So these correlations have this sort of transitive property, which means that because there's correlation between E and K and D, we're also going to see a correlation between E and D, and actually between all of these pairs of sequence positions, even though the E and the R aren't actually that close in the 3D protein structure. Right? So we have a problem. We have sort of too many correlations. So essentially, to get around this problem, we're going to construct a probability model where we take into account interactions. Okay, we want to use the observed correlations to constrain the probability model. So we want to find a model that has the observed correlations. We also want to, or we're going to decide to choose the maximum entropy or least constrained model that has that feature. So we want the smallest possible set of interactions that will reproduce the observed correlations. Okay? And it turns out that this model, or the maximum entropy model, has this functional form. So we have basically these linear terms and these pairwise terms. This is basically like an Ising or a Potts model. And so our job now is to, from that data, infer these parameter values. Right? So we'd like to infer the EIJs and the HIs. And if we make a sort of very simple naive mean field approximation, we assume the couplings are relatively small compared to the fields, then we can infer all of those parameters in one single step just by inverting the observed covariance matrix. So this, of course, would be exact if this was a multivariate Gaussian with continuous variables, but these variables are a little bit odd. They're discrete. They take 20, roughly, states. And so this is only approximately true for this system. But we'd really like to actually be able to do this and find out if it's any good. So we're just going to assume that this is reasonable and carry on. We're going to invert these matrices. It turns out these matrices aren't actually invertible. They're generally singular because we don't have that much data. We don't see every single. We have whole columns of zeros. But to get around that, we're just going to perturb them slightly away from 0, right, by adding some pseudo-counts. And it turns out that when we do that, actually, this is the high-scoring pairs found using that probability model. If you look carefully, you can see that the red points generally are falling on top of these gray points, which are contacts in 3D structure. And you can compare it to the mutual information result, which is shown in the lower quadrant of this matrix, where we weren't able to find many of the actual contacts at all. So you can see that even though we use that sort of rough approximation to infer those parameters, we really have done a much better job of being able to predict these contacts just starting from that sequence data. And so when we saw this was kind of exciting. We really hadn't expected this to work that well, and it led to the question, is there enough information here to actually predict the structure of the protein? This protein starts off as a linear polypeptide chain, and then it folds up. And if we know that some number of pairs are in contact, then at some point we have enough information to actually specify the structure. And this looked like a pretty good set to start with. Okay? And so the only way we really knew to test that question was to actually try it. And so we turned to a technique called distance geometry, which is used actually in experimental structure determination, because experimentally people can often find information about the distances between pairs of sites. And so literally we're going to start with this extended polypeptide chain, and then we're going to constrain the distance between the residues in each high-scoring pair. So literally we're going to say that every pair of sequence positions that has a high score, those amino acids will be less than, say, 7 angstroms apart in 3D space. Okay? And then we'll use this technique called distance geometry to basically find 3D configurations that satisfy this set of constraints. It turns out people have already written a nice software package to do that, which is called CNS, which you can freely download online. And so this has made it quite straightforward to test this out. So this package uses distance geometry, as I mentioned, and then a small amount of simulated annealing on top of that to sort of take apart residues that have ended up on top of each other. And when we did this, it turned out that actually our predicted structures look really quite like the crystal structures that people have experimentally solved. So they're not perfect. There are sort of distinct differences between this predicted structure and this experimentally solved structure. You can see that this beta sheet here, this dark blue strand, is not quite the right shape. And yeah, some of the loops in particular, this sort of green loop here, is not in quite the right conformation, but it's really pretty reasonable, and it's about 3 angstroms root mean square deviation from the crystal structure. So when we saw that, we were really quite encouraged, and so we tried a bigger protein. This is RAS, which has 161 residues or 166 residues. It's quite a big protein. And again, you can see there are some differences between our predicted structure here and the experimentally solved crystal structure, but it's a reasonable prediction. For both of these proteins, there's quite a lot of structural data available. I think for RAS, there was something like 15,000 sequences available. So at least this told us that in the case where we had a lot of data, this works really quite well. So here is a sort of set of proteins that we did this for, and in each case, the crystal structure is in gray, and our predicted structure is in purple. And so we were able to apply this quite widely, at least in cases where people knew the crystal structure. So then we turned to a case where the crystal structure hadn't, or looked for cases where the crystal structures hadn't been solved, and a particularly sort of interesting case is transmembrane proteins, because those are very hard to solve experimentally. Right? These proteins are embedded in the membrane, so they're very hydrophobic, and they're difficult to solve experimentally because of that. And this allowed us to ask whether we could do de novo prediction. Right? Could we predict the structures of proteins that people haven't crystallized before and sort of believe that they were accurate? So of course, first we trained on examples where people have solved the structure. This is a beta-2 adrenergic receptor. You can see that this is the contact map overlay. We're doing fairly well at finding these contacts, and this is the resulting crystal structure. And this is a nice result, to be sort of around 3 angstroms RMSD for this large protein. You can see that there's a fair amount of disagreement at the top. But again, this is a case where we had a lot of sequences, and it worked fairly well. So these were blinded, but we knew the answer. But then there was a set of 11 proteins where the structures hadn't been solved, and we felt we were able to, there were enough sequences, and our predictions looked sort of reasonable enough as protein structures that we were able to actually sort of make predictions for those proteins. So these are structures that haven't been solved before, so there's no crystal structure to compare to. So this is all very nice, and it is sort of surprising, as far as I'm concerned, that this works when there, even if we have if we have lots of sequences. Certainly, if there are fewer sequences, the prediction quality gets worse. So just wanted to remind you that, of course, we are just literally trying to find these parameters. We're making this approximation when we find these parameters, and I think it's a very rough approximation. It's not really clear how well it works at all, and I think Simona in the next talk will talk a little bit more about methods to sort of improve this approximation or do a better job of fitting these parameters. But I just wanted to show you the difference between a protein that has a lot of sequences. This is LAC-I, and we had 20,000 sequences for about 360 variables. This is a true positive plot, so this is just a proportion of true positives as you predict more and more, or as you count more and more high-scoring pairs. And you can see that basically this sort of, at least for most of the methods I've shown here, this sort of stays around 80% or 80, 70% for a good while. In contrast, this is LAM-B, where you have sort of a 10th number of sequences for even more variables. And you can see that there's sort of much more disparity between these different methods. These different methods are basically really rather similar to each other. They have different regularizations. But here you can see this falls off much more quickly. Right? So there really is a problem where we don't have that many sequences. And of course, most of the proteins whose structure we'd like to predict, we have very few sequences for. Okay? So for this really to be useful, we'd like it to work in the regime where we have very few sequences, and that's exactly the regime where we have problems. It does seem to sort of clearly be to do with sampling, and we sort of were able to show, I'm sort of flicking over this slightly quickly, that if we use shrinkage, so instead of just using the covariance matrix, we basically upweight the diagonal, then we can do significantly better. So this is with 0 shrinkage, our true positive rate for 100 pairs is around 60%. But then if we use shrinkage, we increase the shrinkage intensity away from 0, you see that it gets much better. It goes up above 80%, sort of almost approaches 90%. So that suggests that it really is a sampling issue. We're rather undersampled. So this led us to sort of ask, are there other ways we could use to sort of tackle this sampling problem, right? And in particular, could we sort of turn to random matrix theory and use spectral analysis to distinguish between signal and noise? So I'm going to sort of turn to a simple mathematical model in order to explore this. I'm going to consider, instead of having 20 amino acids, I'll just have 2 amino acids, a binary system. And I'm literally just going to start with a binary string of length, say, I think in this case 160, and I'm going to inflict mutations on that binary string, right? So let's say I make many copies of my initial sequence, and then I mutate each one 1,000 times, so many, many times. So I'm going to produce a data matrix where each row is a sequence, and each column, obviously, is one of the sequence positions. And then I can calculate the covariance matrix, either x prime x or xx prime, of that data. And I can sort of ask, well, what's the empirical eigenvalue distribution? Well, of course, if I mutate every sequence 1,000 times, every site should get hit many times, so they should end up being totally independent, right? Random matrix theory tells us that the eigenvalue distribution of the sample covariance matrix sort of has a characteristic shape. And in particular, if the true covariance matrix is the identity, then we have the Marchenko-Pasteur law. Of course, other models of the true covariance matrix give different distributions, and often we can compute these using the Marchenko-Pasteur equation, which we'll talk about a bit more later. But for now, for that system I showed you just now, this green is a Marchenko-Pasteur law, and the blue is the observed spectrum. And you can see that we really have quite a nice fit between the two. Okay, so far so good. As long as our sequences are sampled independently of each other, as long as they're IID, right, then we know what the spectrum should look like. So if we add some interactions to that system, if we put interactions between the variables, the sequence positions, right, then what effect does that have? So I'm going to sort of continue to just use Marchenko-Pasteur, the Marchenko-Pasteur distribution, because my sequences are independently sampled. But now you can see that I have some density or some eigenvalues outside, some discrete eigenvalues outside of this Marchenko-Pasteur bulk. And so this is sort of interesting because it suggests that maybe we can find out something about the interactions just by looking at these modes. Okay, so that's sort of an interesting direction. So if we're really undersampled, maybe we can just use this approach for our data. So there's a couple of issues with this model. And the first is that, well, of course, our sample sequences aren't actually independent of one another, right? They're related by phylogeny, by the evolutionary process. And so really, we'd like to understand, if we're going to use this approach, how the parameters of the tree affect the eigenvalues of the covariance matrix. Is it still roughly Marchenko-Pasteur, or does it look quite different? And I also sort of assume that the signal was low rank. And if it's not, that was an assumption, does it still result in these sort of isolated eigenvalues that detach from the bulk? So first I'm going to talk about phylogeny. I guess I probably won't have time to talk about the signal rank, but anyway. So we have this simple mathematical model, this simulation. So let's consider, instead of having all the sequences be independent, we're going to enforce a phylogeny. And we'll do that by literally starting with some number of founder sequences, mutating them, and then when we get to a branch point, we will duplicate all of the sequences that we have. So half will go this way and half this way, and then we'll mutate all of our sequences for a further period of time independently of one another, and then we'll duplicate. And so we'll end up generating some large number n sequences. Turns out if you then make the sequence covariance matrix, it sort of looks something like this, at least for a regular branching tree. This tree needs to be a little bit bigger. But you can see there's really quite a lot of structure in this sequence covariance matrix. And this is sort of slightly worrying because it no longer looks like a sort of Gaussian, the covariance matrix of a Gaussian with an identity covariance matrix. Well, of course, this is a very simple Markov chain, so we can actually compute the eigenvalues of the true covariance matrix. So if we define this parameter alpha, which is basically depends on the number of mutations per branch and the length, the number of variables, then the elements of the true covariance matrix are given by this straightforward function of alpha, and the true covariance matrix basically looks like this. So if you look at that sample covariance matrix from the slide before, you can see it sort of has roughly that structure. There are these blocks which are basically different powers of alpha in this very regular case. So we can write down the eigenvalues of this true covariance matrix, and then we can ask what's the spectrum of the sample covariance matrix drawn from that distribution. Of course, if we make M large enough, if we inflict enough mutations on those sequences, then they'll still end up being independent, right? So we'll still recover Marchenko-Pasteur. But in that case, our sequences basically look totally different from each other, and we know that's not the case in biology, right? That's definitely not true. So if we inflict fewer mutations, if we say choose M equals 45, then this observed bulk, or our observed spectrum, looks really quite different from this green dotted Marchenko-Pasteur. And if we make M even smaller, then we find that these bulks have actually separated. Right? So if we're comparing our data to Marchenko-Pasteur, and we're actually in this regime, then we're sort of working with the wrong model. Now, you might notice that I've also put these red analytical curves on these plots. And those come, as I mentioned earlier, from solving the Marchenko-Pasteur equation, because these systems have discrete eigenvalues, so we can literally just, well, the Silch's transform of the empirical spectral distribution satisfies a polynomial equation, and we can just find the roots and use those to put the analytical distribution on top of our data, and you can see that it really matches quite nicely. So it's nice because we actually sort of have a theory of what's going on with these distributions. And indeed, you can see that as we change this parameter alpha, if we start from Marchenko-Pasteur, the bottom of the edge of this bulk moves outwards, following this red line. So the red line is found by using Sturm's theorem, essentially to solve for the edge of the bulk as a function of the parameter alpha. And on top of that, I sort of carried out lots of simulations, or I have a really good graduate student who's carried out lots of simulations. You can see they fall very nicely on top of this red line. And just for comparison, I put the Marchenko-Pasteur edge in black on this plot, and then I put the edge that would be found if we constructed a Marchenko-Pasteur distribution with one of the eigenvalues of this matrix. So basically, this matrix has two eigenvalues, 1 plus alpha and 1 minus alpha, and the green dashed line shows the 1 minus alpha edge of the bulk. And you can see that it's sort of, I don't know, between those two extremes. And again, the same is true for the upper bound of that bulk. You can see the simulations really match that analytical solution rather nicely. And of course, we also have this phenomenon where these two bulks split apart. And again, using Sturm's theorem, we can solve for how this critical value at which they split changes as a function of these parameters alpha, and this is the concentration, so it's a ratio of the number of variables to the number of samples. And so we can see that essentially, this critical value that is split apart basically seems to look something like a square root. So we have at least some understanding of what's going on in this model, and then we can extend this to more complicated trees. So, if we have 3 branching events, it can look like Marchenko-Pasteur, but we can also have this very bizarre distribution where we almost have 3 or 4 bulks. The spectrum is really split up into different pieces. And here, it's sort of just extending beyond Marchenko-Pasteur. And so basically, I think what we learn from this is that correlations between samples significantly alter the empirical spectral distribution in the absence of any signal. And so that makes it very hard to identify signal modes from data, at least unless we know exactly the tree that the samples, or the relationships between the samples that we have. And the problem, of course, with trying to infer that from the data is that we'd be using the correlation between the samples to infer that, and we're sort of, that's what we're trying to analyze. So it's sort of a bit tricky. I mean, there are ways of trying to get the phylogeny without just using the sequences you have by looking at other proteins from the same species. And so it might be possible to do something with that. Okay, so essentially, I think I've shown that sequence covariation can give accurate prediction of protein structure, at least if we have enough sequences. But I think there are a lot of outstanding questions. not only sort of pragmatic questions, if we're going to use this for prediction, along the lines of, how many sequences do we need, how different from each other do they have to be, and like what are the expected errors in predicted contacts, and how do they depend on the data that we have. We'd really like to actually understand this, rather than just being able to sort of make predictions and, see how well they work in different cases. I showed you a very simple toy model, but it's very unclear how strong the correlation should be. I just put ones and zeros in my toy model, and I added those interactions, but we don't really have a good idea about these interactions, their strength, or really even what they reflect. I've sort of presented an approach to addressing some of these questions, which uses random matrix theory. I think it's true that if the correlations were, or the interactions were low rank, or the signal was low rank, then there's sort of a clear mathematical framework. This phylogenetic relatedness changes the shape of the underlying distribution, but if we can get a handle on that, we can actually sort of say what the phylogeny of a particular data set is, then it seems like we might be able to understand or specify the expected sort of empirical spectral distribution, and so use that approach profitably. And so this is something that we're currently working on, to apply this to real phylogenies. But I guess the problem is that I'm not actually really sure that it's clear that the correlation or the interactions are a low-rank signal. And of course, if they're not a low-rank signal, it means they could change the shape of the underlying spectrum, which is sort of worrying. So I think basically we need models that are accurate to help us deal with all of this data. And I need to thank the many people that I've been lucky enough to collaborate with. This is Chong Li, who's an excellent graduate student who's done the random matrix theory bit of this work, and these are some of the people who have funded us. And thank you very much for listening. Thanks.